I am Krista Moore, and I'm going to be introducing our speaker for the Cell Bio Lab. It's Shani Siegel, and she's going to be presenting her work that actually ties in really nicely, thinking about DNA damage and how we can think about these genes and drug interactions. So I'm going to let her get started here, and she's going to start sharing some slides.
Awesome. I will share my screen. All right. Good evening. First off, thank you, Dr. Moore, for that introduction. My name is Shani Siegel, and I'm the Cure Symposium speaker for MCDB 3140. And throughout the semester, I was looking at the gene fatty acid 2 hydroxylase, also known as FA2H, to hopefully find another gene involved in the DNA damage response pathway. So to get us started, I wanted to bring to your attention why the DNA damage response pathway, or DDR pathways, are incredibly important to the promotion of cancer research. As we know, organisms are constantly exposed to DNA damaging reagents, so it's necessary for us to have functioning DDR pathways to live. But while these processes are crucial to life, diseases can take advantage of the misregulation of these pathways. And so it's already been shown that many cancers are the result of disrupted DNA repair pathways resulting in chemotherapy and radiation resistance. Yet with a better understanding of the current components in DNA repair pathways, cancer and other diseases can be targeted with more effective treatment strategies by manipulating the regulation points of these pathways. And experiments have already shown that by decreasing DDR factors, we're improving the therapeutic effectiveness of current cancer therapies because the cells cannot repair DNA damage. Yet many of the genes and components of these pathways are still unknown, yielding the opportunity for better control over these pathways in the future. And so in order to study these pathways of DNA damage repair, we use Tetrahymena thermophila as our model organism. So Tetrahymena are unicellular organisms that not only are easy to grow, but they also undergo immense DNA rearrangements during sexual conjugation that leads to significant DNA damage and repair. So they are excellent model organisms to view the effects of gene expression after the imposition of DNA damaging chemicals in an in vivo experiment. So the gene that I was looking at this semester was FA2H. This gene has 3 exons shown here in blue and 2 introns shown here in black. And FA2H has a human homolog as well as a possible functionality of cholesterol and fatty acid biosynthesis. So clearly, this gene does not have functions that would indicate that's involved in DDR and overall appears rather ordinary for a gene. So why was FA2H selected out of the supposed 27,000 predicted protein coding genes in the tetrahymenic genome? Well, it was chosen based on its gene expression profile. So FA2H has a similar gene expression profile pattern to genes that are known to play a role in DDR, such as RAD51 and HOP3, both shown here. And after the beginning of conjugation, FA2H also exhibits that increase, decrease, increase expression pattern, similar to what's observed for genes known to be involved in DDR. So due to the similarity of the gene expression profiles, FA2H was selected to be studied as a potential candidate for a gene that is involved in DDR and is also upregulated during the conjugation in Tetrahymena. So the central question of this project was, does inducing DNA damage cause the upregulation of FA2H, indicating that this gene could be a part of the DDR pathway? To answer this question, cells were extracted from Tetrahymena, and one sample was exposed to the DNA damaging reagent hydroxyurea, and the other half was left untreated. And so incubation with high concentrations of hydroxyurea can result in cell death and DNA damage generated at arrested replication forks. And then the RNA was extracted from both samples, and then using GoScript reverse transcriptase, cDNA was made, which was then amplified using an RT-PCR. With an RT-PCR, if a gene is expressed, then mRNA is going to be made. And the more RNA that is made, there's going to be more amplification, which will show up as a significantly thicker and brighter band on the gel. And so basically more expression correlates with more amplification since more mRNA is made. And then once the gel is run, we're going to get these bands, and the larger band would indicate that there was more cDNA, which is what I was expecting due to the gene expression profile of FA2H, versus the untreated sample, which should have significantly lower brightness. And then these bands would also be ran against a ladder for measurement purposes. But before I would even be able to run a gel or reverse transcribe the RNA to determine if FA2H is in fact involved in DDR, I needed to design and validate my primers. So with these primers, they needed to anneal specifically to Fa2H and will also amplify Fa2H in PCR. And the expected primers blank an intron, resulting in a difference of size between the gDNA and the cDNA. And the expected length for the gDNA was 617 base pairs, and the expected length of the cDNA was 544 base pairs, excluding that intron, which ended up making analysis on the gel much easier since the bands would appear at different sizes. So the primers were designed, and then I isolated the cDNA and the gDNA with these primers, and then we ran a PCR, and then the PCR products from the primer validation experiment were run on a gel electrophoresis. So this gel was looking at if the primer specifically annealed to Fa2H and could be used in further experiments. This validation was necessary to ensure that the results that I viewed on the expression analysis gel were due to DNA damage and changes in expression as opposed to the primers not annealing. And so this allows me to know that if there's no bands for the expression gel, it's because there was no expression and not because the primers didn't anneal. So what I did to validate these primers was I used a 1 kb plus ruler here to better see the distinct sizes of these bands, since there was a good spread of expected sizes. And I observed with Fa2H the 544 base pair cDNA, as well as the 617 base pair gDNA, which was the expected length for both. And then to ensure that my data was reliable, I ran a positive control with FTT18, which had the expected sizes of 1500 base pairs for the gDNA and 550 base pairs for the cDNA. And since I had primers that we knew annealed specifically to FTT18, this was really good for ensuring that my reagents and overall PCR setup were valid. And then the negative control rows right here had no DNA template and also showed no contamination, indicated by the fact that there were no bands in those rows. And so as this data matched what I was expecting to observe with the lengths and no contamination, I can conclude that my primers annealed specifically to Fa2H and then can also be used in further experiments. So the primers were validated, DNA damage was induced, RNA was extracted, cDNA was made and amplified, and then the last experiment was completed and the last gel electrophoresis was run. So to examine the expression changes of Fa2H after DNA damage to determine if this gene is actually involved in DDR pathways, I ran another gel using the reverse transcribed cDNA and specific primers of RAD51, FTT18, and Fa2H. And so the purpose of using reverse transcribed cDNA was to examine expression, since not all DNA is expressed at any given time. And this would indicate that if I see no band, then Fa2H was actually expressed in the cell and was not the result of the gene simply being included in the genome. And so for this gel, I used a 100 base pair ladder, since all the bands were expected around a very similar length. And we can observe with my first positive control RAD51 that it was not only the expected lengths of 720 base pairs, but the gene expression significantly increased after DNA damage was induced, shown here in column 3. And this just indicates that DNA damage was also induced throughout the entire experiment. And it also gave me a guideline for what I was expecting with Fa2H, as I had thought originally that Fa2H expression would be significantly upregulated after DNA damage was induced, resulting in a brighter band like this. And then with my second positive control, there likely was some experimental error, as FTT18 is constitutive and it should have been expressed evenly with or without DNA damage. But as we can see, there was a little bit of variance, but nonetheless, it wasn't drastic enough to invalidate the experimental setup. And then also just to verify that there was no contamination that could have influenced the results I viewed, in lanes 8 through 10, I used a -RT control, meaning that I did not include any reverse transcriptase enzyme. And so I was expecting no bands here as well. And lastly, with Fa2H, I saw in the DNA damage induced column that there was not the bright band that I was expecting, but instead there was a significantly less bright band, and that's demonstrated right here. And so from this information, I can conclude that Fa2H is instead downregulated after DNA damage was induced and not upregulated like I was expecting. And since my FTT18 did not look as constitutively expressed as I hoped, I did a quantitative analysis normalizing to removing background frequency instead of normalizing to FTT18. And I still saw that RAD51 was expressed approximately 325% more in treated samples versus untreated samples, and Fa2H was expressed 86% less in treated samples versus untreated samples. So overall, based on the data that I have, I can conclude that Fa2H is downregulated after DNA damage. And this initially led me to think that FA2H would then not play a role in DDR. But as I analyzed these results, I really began to believe that this data doesn't really indicate that at all. These results only show that FA2H is not involved in turning on DDR and the immediate response pathway to DNA damage, but it doesn't indicate how FA2H interacts with other components of the DDR pathways, nor later advancement within this pathway. So through FA2H's interactions, it's really possible that it could be absolutely necessary for DDR to occur, but yet this was not demonstrated from this data. And as I stated before, current studies have already demonstrated the effectiveness of decreasing DDR factors with increasing the therapeutic effectiveness of current cancer therapies. And so by finding more genes that are involved in DDR pathways, there's going to be more targets for gene therapy to alter these expression patterns and turn off the DDR pathways and make these traditional cancer therapies more effective. And FA2H has an incredibly similar human homolog, and so this makes it even more necessary to further examine FA2H's function in DDR, as this gene could serve as a potential target in human DDR pathways if it is involved. And lastly, gaining more knowledge about how FA2H affects DDR indirectly, or if this gene is involved in turning this pathway off, is a possible aspiration for better understanding the role of FA2H in DDR. And so I think that one of the most important next steps would be to upregulate FA2H in cells that were exposed to DNA damaging reagents, and we can observe if the cells were still able to repair their DNA. We could also use X-rays to induce double-strand breaks and see if expression is similar to what I currently observed. I also thought it would be really interesting to look at FA2H's protein interactions and observe if it interacts with proteins that are also involved in DDR. And then we could also use drugs that induce double-strand versus single-strand breaks and see if FA2H's expression varies between the two types of damages. But lastly, we could also simply say that based on the information I have, it would appear as though FA2H is not a significant driver in DDR and look for other genes that may play a more significant or direct role. But I do believe that this should be the last resort because I think there's a lot of knowledge to be gained with FA2H and it would be really promising since it has a very similar human homolog. Firstly, I would like to thank Dr. Moore, Stella Koliavas, and Carrie McMillan for their unconditional support throughout this project, as well as the MCDB Department and University of Colorado Boulder for their support and funding of this endeavor. And here's my references. I am now open for questions.
All right, great job. Any questions?
I could ask a question. Great job, Shay. I was just wondering for the hydroxyurea, if it does kill the cell instead of damage it, like if you use a really high dose, could that be an issue? Or no, because you're eventually going to kill the cell anyways to get the RNA from it?
That's a good question. All I really know about hydroxyurea is that it kind of like arrests the replication fork, which just causes like a break, but that's where the DNA repair factors are going to come in. So I don't know how often it's going to directly kill the cell unless it's at a really, really high concentration. But I think we probably use concentrations that would kind of just induce the DNA damage at the point where DDR could still turn on before the cell died.
That makes sense. Thank you.
Of course.
Great. It looks like we've got some questions in the chat. Jillian, go for it.
Yeah, I was wondering, so if you suspected that it still potentially plays a role in that DNA repair pathway, why wouldn't it be like constantly expressed and like it was downregulated instead? So if it were important to that pathway, why was it downregulated? Like why would expression actually decrease?
Yeah, so kind of what I was thinking as like a possible path for this would be its downregulation is absolutely necessary for another protein to be activated that is a crucial player in DDR. And so by downregulating FA2H, it's possible that then could stimulate the entire pathway. So that's why I thought it would be really cool to upregulate it deliberately. And then we could see if DNA repair pathways are still able to progress. Because then we know that the downregulation is absolutely necessary for the repair pathways to continue.
Awesome. And I think maybe we have time for one more. So Ben, if you have a question.
Yeah, I have one question, has to do with your future directions, and were you wanting to look into basically whether it was a double or a single break in like the genetic sequence? And I was wondering like what sort of factors can affect whether it's a double or a single break, and what you almost like predict your research would do, like whether it'd be effective or how it would like, yeah, how it'd be affected by whether it's single or double.
Yeah, so kind of what I was thinking with that is there's different pathways that are going to go into whether it's a double strand break and repairing that versus just repairing a single strand break. So while it might not be that FA2H is upregulated in one of those, it could be upregulated in another one. So I thought it would be interesting to be able to compare the two different pathways and see if it's involved in one of those, because they're kind of completely different pathways and they're going to have different enzymes and proteins that are going to be affecting them. So yeah, good question.
Awesome. Well, unfortunately, I think we're out of time. Looks like there might be some questions in the chat, and if anybody else had one, feel free to throw it in there. She might be able to answer for you. And great job. And I will hand this off, I believe, to Symbio.